Hello friends, my name is Mr. Kiran Dhawale, Department of Chemistry, RBNB College, Sri Rampur. Today we are going to discuss about syllabus of Analytical Chemistry CH202. This is the subject code, and that subject Analytical Chemistry, which is for FY/BSc semester 2nd, which is in the new pattern CBCS CH202 Analytical Chemistry and have 2 credits okay, and near about 36 lectures are allotted to this whole syllabus for one semester. Okay, then it contains 5 chapters. First chapter is Introduction to Analytical Chemistry. In this case, that chapter is giving some idea about analytical chemistry. Why? Because that subject is introduced firstly in this syllabus of FY/BSc. Before that, there is only a physical chemistry, inorganic chemistry, and organic chemistry. That study is already learned by the student in 11th and 12th class, but in case of FY/BSc first year BSc, in that case this is the new subject for that student. So here first chapter is Introduction to Analytical Chemistry, which gives some idea regarding and basic knowledge regarding the analytical chemistry. So what type of topics which included in that chapter? This is of 3 lectures. The chapter has 3 lectures, and near about whole syllabus is about 36 lectures and 35 marks. Total marks for this course or for this subject is 50, but in University of Pune, that 50 marks can be divided into 2 parts: university paper and for internal evaluation. For university section, the student will exempted, or which will give exam, a theory exam of this syllabus of about 35 marks, and remaining 15 marks having internal evaluation, which contain some internal assignments. Then it's a semester overall performance, presentee, and it's one type of module which will fill by the student in that case. And here total actual syllabus is of 50 marks, but having 36 lecture, which can be divided into the 5 topics. And first topic is Introduction to Analytical Chemistry, and is of near about 3 lectures. The next one, second chapter, is Calculations Used in Analytical Chemistry. So this is the second chapter. Which has 10 lectures. Then third chapter having Qualitative Analysis of Organic Compounds. qualitative analysis of organic compounds, and has 5 lectures. Then third or fourth chapter is chromatographic technique. Chromatographic techniques, which contain paper and thin layer chromatography, and this chapter have 14 lectures. So this is one of the biggest chapter in the syllabus. Then last chapter is pH meter. pH meter, and have only 4 lectures. So first and last chapter both have near about equal weightage. Here first chapter, which is only of introduction about analytical chemistry, then second chapter which contain different type of calculations we used in analytical chemistry. If we carry out any experiment, at that time, at the end of that experiment, there is explanation of or gathering some data which is getting from that instrumentation or that practical. At that time we can gather that data and make some solutions by using some formulas and make up to its conclusion, so that what type of calculations we use in analytical chemistry, that is discussed in that chapter. Then third chapter is qualitative analysis of organic compounds. In that case we learn regarding what type of test we taken. In this case, when we take an organic compound, either that organic compound in binary phase or ternary phase, when two compounds we mix with each other, and at that time how we can identify and how we can separate them by using different literature or by using different methods, which can be discussed in deep in this syllabi. How we can filter, what is meant by filtration, then what are the types of filtration, then what test we can carry out, what elements which we can detect, and what type of element, if we can find out the if your compound contain nitrogen, sulfur or halogen, either that is present or not, so which test we use for detection of that. So Lassagne test, or we say here this is sodium fusion test. Okay, and what is the actual procedure for that? What are the simple test for detection of that nitrogen, sulfur and halogen? So these are totally discussed in that syllabus. Then next, what are the techniques we used for the purification? Either we use, it can depends on the what type of compounds we use for the purification, how we can increase its purity, and what methods we use for that. So recrystallization, distillation, then sublimation, these are some topics we discussed or we take as a practical. Then next chapter, which is chromatographic technique, which contain, we focused on the paper chromatography and thin layer chromatography, especially these two topics we discuss in deep. But instead of that, we take some principle or overview of other type of techniques which used in chromatography, such as gel partition, exchange, ion exchange chromatography, affinity chromatography, gas chromatography, supercritical fluid chromatography, high performance liquid chromatography, which is also abbreviated in HPLC, you already known or you listen that word. Then capillary electrophoresis, then it's a classification, and these two chromatographic techniques we discussed in brief in this section, paper chromatography and thin layer chromatography. The next and last chapter, which is pH meter. pH meter, which contains some introductory part, then what type of pH meter we used and why that used, what are the principles of that, then combination of pH electrodes, then complete cell, standard buffer. The next one, accuracy of pH measurements using pH meter, how does it work and what are the applications of the pH meter. So this is the whole syllabus which we discussed in this semester. So here all over these are the 5 chapters. In that 5 chapters, 2 chapters are very important for us, and more weightage, near about 60% weightage is only of these 2 chapters, and which are the calculation used in analytical chemistry, which is have 10 lectures. And next one is chromatographic technique, which is the first largest chapter in this case, here having 14 lectures, so which is the major part of this syllabus. Then next to that, I think this one is qualitative analysis of organic compound, having only 5 lectures, and that will be discussed either not in case of theoretical case, we discuss in that practical laboratory. So such a type of techniques we understood very well by using some practical techniques. Okay. The next one is this one, pH meter, and last is the introduction to analytical chemistry, but this is important for us. We doesn't think about its mark, but we think about its basic knowledge of analytical chemistry. So we start with the topic introduction to the analytical chemistry in next lecture. Thank you.